Hello and welcome to EduSathi. We'll be studying biology today. So let's get started and start with cell. Now, cell is the basic structural, functional, and biological unit of life. Cell is from where the life starts. Now, this term cell was coined by the British scientist Robert Hooke in the year 1665. He was the one to discover cell. The smallest cell that we have is Mycoplasma gallisepticum. The largest one is neuron. The biggest cell is the egg of ostrich. Sheldon and Chavan established the cell theory during the years 1838 and 39. So in this slide, I have to remember the name of Robert Hooke, who discovered the cell, the smallest, longest, and the biggest cells, and Sheldon and Chavan, who established the cell theory in the year 1838-39. Now, cells are of two kinds, prokaryotic cell and eukaryotic cell. So you can just refer to this figure while I'm talking about prokaryotic cell. Prokaryotic cells are single cell with nuclear material but no nuclear membrane. So there's no nuclear membrane that we have. The nuclear material which is present is just spread in the region. Okay, and the other membrane bound organelles are also absent. Other membrane bound organelles such as mitochondria, ribosome, Golgi bodies, all of them are absent in a prokaryotic cell. So prokaryotic cell is a very small single cell that we have where there is no nucleus. So there is no nucleus that we have. The nuclear material that is there is just spread in it. And we have a naked DNA. So there is no nucleus and it contains single chromosome only. So I have a single chromosome in a prokaryotic cell. The membrane bound organelles are not there, and the cell division takes place either by fission or by budding. So fission or budding are the ways in which the cell can divide. Okay. Now let's look at the eukaryotic cell. Now eukaryotic cell, if I talk about, they are the complete cell with organized nucleus membrane and other membrane bound organelles. So I have a very organized nucleus that you have. I have very organized other membranes such as Golgi bodies, mitochondria, ribosome, all of them are there. So if I talk about this cell, this is relatively large in size as compared to a prokaryotic cell, and there is obviously nucleus is present. All of them are in very organized manner. It contains more than one chromosome. More than one chromosome. There was only one chromosome which is present in prokaryotic cell. There are more than one chromosome which are present in eukaryotic cells. Okay. Obviously, there are cell organelles which are present. Organelles. We'll talk about cell organelles in detail in the following slide. So, there are cell organelles which are present, and the cell division happens by mitosis or meiosis. So, these are the two ways in which the cell division can take place, mitosis and meiosis. Okay. Now, if I talk about the examples, now virus, bacteria, and cyanobacteria, CYNO, cyanobacteria are some of the examples of prokaryotes. Okay. All the unicellular and multicellular plant and animal cells, they are eukaryotic in nature. So all the unicellular or multicellular, the unicellular means one cell, multicellular means many cells. Animal as well as plant cells, they are all eukaryotic in nature. So these are primitive cells. Prokaryotic are primitive cells. They are modern day cells. Most of the cells are eukaryotic in nature with very organized nucleus and membrane bound organelles. I hope there's no confusion that you have with respect to prokaryotic and eukaryotic cells. Okay. Now let's look at a eukaryotic animal cell. Now, if you just look at this cell has an outer membrane. It has some internal organelles. We'll talk about all of these internal organelles one by one. So just keep this figure in your mind And let's move ahead to the next slide. In this slide, we'll be talking about all of the cell organelles that we have. They're also known as factory components with a specified function. So all the cell organelles that we have carry out some specific functions inside the cell. Now let's talk about the function. Support. Cell wall, cell membrane and microtubules provide the support and rigidity to the cell. Now this is what the outer covering is there. So this is the cell membrane that we have, also known we have a cell wall in case of a plant cell. If I talk about an animal cell, there's no cell wall that we have. We have is cell membrane. It is also known as a plasma membrane, which is the outer covering of the animal cell. Now in a plant cell, I have a cell wall outside the cell membrane, but in an animal cell, what I have is the cell membrane. Now this is what provides support, rigidity to the cell. And it also it is a kind of a permeable thing, so it allows inward and outward movements of material. So cell membrane, apart from providing support, also controls the material entering and leaving. So the cell pores also are there for the same purpose. They control the material entering and leaving the cell. Now once the material has entered inside the cell, now there is a membrane network of tubules which internally transports them, and they are known as endoplasmic reticulum, also known as ER. So it's a network of tubules which is found inside. This is what you can look at. This is a kind of endoplasmic reticulum that we have, and this is what endoplasmic reticulum look like. So I'll just change the color of the pen so that you can just look it. This is what the endoplasmic reticulum looks like. So this is all endoplasmic reticulum. It's a structure which maintains the internal transport system. Okay. So it helps in the distribution of material, and also helps in supporting the framework of the cell. Okay, now the next one that you could see, this is mitochondria. Now what does mitochondria do? Mitochondria is known as the powerhouse of the cell. Powerhouse of the cell, that means the power is generated inside the mitochondria. So if I talk about, it's the respiratory site of cellular respiration. So it's basically where the respiration takes place. This is what the internal structure of mitochondria looks like. Mitochondria has an inward internal membrane which forms some kind of protrusions which are known as cristae. So it is a site of, now this mitochondria is a site of cellular respiration, and it releases an energy rich compound which is known as ATP. And because of the energy which is generated here, it is known as the powerhouse of the cell, which is mitochondria. Okay. Now, if I talk about the control center, the one which controls the cells is the nucleus. So nucleus is the one which controls the cell, and it's the basically the you just look at the structure that we have, this one, the round structure that we have. It is kind of spherical, centrally located, and is a major structure found inside the cell. It controls all the activities of the cell. It is the control center. So it is the one which controls all the activities inside the cell. Okay. Production of key products is done by ribosomes, endoplasmic reticulum, and chloroplast. Now, if I talk about what is ribosome, I've talked about endoplasmic reticulum also already. If I talk about ribosome are the things which are made up of RNA, or I should just say ribonucleic acid. So they are made up of ribonucleic acid, and they take part in protein synthesis. So they synthesize the key products inside the cell. So they take part in protein synthesis. If you want to look at the ribosome, they're just spherical things which are present inside the cell. Okay. Endoplasmic reticulum also helps in transport, and it also help in synthesizing the key products inside the cell. Okay. Packaging center for shipment of products is Golgi apparatus and endoplasmic reticulum. Now, what is Golgi apparatus? It's basically a group of tubes, vesicles, and vacuoles, and they work for storage, processing, and packaging of material. So all the storing, packaging of the material which has been produced is done in Golgi apparatus. This is what it looks like. So it's a kind of network of tubes, vesicles, and vacuoles, and it is also involved in the synthesis of cell wall, plasma membrane, and lysosomes. So this is basically a packaging center for the shipment of the products. Okay, shipment of materials out of the cell. Now I have to ship them out of the cell. Golgi apparatus and vesicles will help me in shipping the material out of the cell. Okay, we have just talked about Golgi apparatus. It's the packaging center for the shipment of products, and it ships the material out of the cell. For storage, vesicles, vacuoles, all of them are used for storage of liquids and solids inside the cell. Okay. The next one that we have is recycling center. As the name suggests, uh, this helps in recycling of the material, and it helps in intracellular digestion. So the organelles, lysosomes, and peroxisomes, they help in intracellular digestion. The enzyme which is found inside lysosome may digest the entire cell. So it has the ability to digest the entire cell. It is also known as the suicidal bag. DAL, suicidal bag. So, lysosome are also known as the suicidal bags of the cell. Okay. The next one is convert light energy into chemical energy. Chloroplasts are the one which convert the light energy into the chemical energy. The next one is allows new cell factories to be produced. The nuclear material, which is the DNA, allows the new cell factories to be produced. It helps in the cell division by the process of meiosis and mitosis. The centrioles and the cell wall, it allows all of these things. Okay. Now, let's talk about it in a reverse way. Now we'll now talk about the different components that we have and the main function that they carry. So we have already talked about some of them. We'll just revise them, and the one that we have not talked about, we'll talk about them in detail. So the first one we'll be talking about is cytosol. Now cytosol contains many metabolic pathway. It helps in the protein synthesis as well. Nucleus is the main center of the cell. It controls all the activities of the cell. It is the control center that we have. It contains the main genome, and it synthesizes the RNA and DNA. RNA is a ribonucleic acid, whereas DNA is deoxyribonucleic acid. So it synthesizes that, and it also transmits the hereditary characters from parents to the offsprings. Okay. The next one that we have is endoplasmic reticulum, which is ER. We have already talked about that it helps in the transport of the material inside the cell, and apart from that, it's also help in the synthesis of the key elements which of the cell, and it helps in the synthesis of lipids, proteins, and for distribution to the different organelles and to the plasma membrane. The next that we have is Golgi bodies, also known as Golgi apparatus. Now this is a group of a group of tubes, vesicles, and vacuoles, as I've already told you. Now they work for storage, processing, and packaging of the materials. Okay, and all the packaging, sorting of proteins and lipids, which is there, is carried out by Golgi apparatus. Lysosome. Now, lysosome helps in intracellular degradation or intracellular digestion, and it is also known as the suicidal bag. Talking about endosomes, they help in the sorting of endocystoid material. Moving on, mitochondria. Mitochondria is also known as the powerhouse of the cell and is the respiratory site for cellular respiration. Mitochondria synthesize energy-rich compound ATP by oxidative phosphorylation, and it's a very energy-rich compound which helps in making it the powerhouse of the cell. Moving forward, we'll talk about chloroplasts. Now, this chloroplasts are only present in the plant cell. They are the green pigment which is found in the plant cell, and they help in the process of photosynthesis. So, this is also known as the kitchen of the cell. So I can just say the kitchen of the cell is the chloroplast, and chloroplast helps in ATP synthesis and carbon fixation by the process of photosynthesis. Okay. The next one that we have is peroxisomes. Now peroxisomes do the oxidation of the toxic molecules which are present inside the cell. Okay. Now let's look at the plant cell. Now there's a difference that we have in the plant cell with respect to the animal cell. Now if I talk about the plant cell, now plant cells are larger in size as compared to the animal cell. So the first thing is plant cells are large in size in compared to the animal cell. Cell wall is present in the plant cell which is not present in the animal cells. Okay. Next, there are some plastids that are present. Let me just write it out. Plastid, which is present in the animal cell, but it is absent. Sorry, which is present in the plant cell, but it is absent in the animal cell. Vacuoles are larger inside. Vacuoles are larger in size in plant cell rather than in an animal cell. In an animal cell, the vacuoles are smaller in size. Okay. Now these vacuoles stores the toxic metabolic waste. It's like a dustbin that we have. It stores all the toxic metabolic wastes. Okay. So let's look at the plant cell. We have all the main organelles. Apart from them, we have extra. So we have Golgi bodies, Golgi vesicles, Golgi apparatus. We have endoplasmic reticulum, which helps in lipid synthesis and which also helps in the transport. We have nucleus. We have again the endoplasmic reticulum. We have these mitochondria. Mitochondria are there, which are powerhouse of the cell. That we have cytoplasm all around. We have chloroplast, and the other such organelles that we have. So if I talk about the plant cell special features which differentiate it from the animal cell, is the cell wall, which helps in the protection and support of the plant cell. There are chloroplasts which are there for the photosynthesis, which are absent in an animal cell. There's a large central vacuole that we have for storage and increased surface area. Okay. So this is what the basic difference is there between the plant cell and the animal cell. And both of them do the cell division either by meiosis and mitosis. Okay. The next subtopic that we'll be discussing in biology is ecology. Ecology is the study of interrelationship between living organisms and their environment. Let's look at this figure. Now, if I talk about an ecosystem, ecosystem can be divided into two parts. Now, first of all, what is ecosystem? Now, this ecosystem or the ecological system was the term which was first coined by the scientist named Tansley. Okay, every ecosystem is made up of two components, the biotic component, which is the living part, and the abiotic component, which is the non-living part. So if I'm talking about the relationship between the living organisms and the environment, now these living organisms would be the part of biotic component, and the non-living ones, which is the part of the environment, would be a part of abiotic component. The biotic component, which is the living part, can be further divided into three parts, and they are producer, consumer, and decomposer. So if I talk about producer, those who can make their own food are categorized as producer. For example, green plants. Consumer, those who consumes the food made by the others. So if plant is making the food, so if the food is consumed by the consumer. And the third one we have is decomposer. Decomposer are the ones who decomposes the dead producers and consumers and changes them into physical elements. So decomposers are the one that work upon dead consumers and dead producers and they convert them into physical elements. Mainly, if I talk about fungi and bacteria are counted in decomposers. So all the living things can be categorized either as producer, which is who make their own food, consumer who do not make their own food but consumes the food which is produced by the producer, and decomposer which act on dead consumers and dead producers to convert them back into physical elements. Now, consumer further are of three types: primary consumer, secondary consumer, or tertiary consumer. Primary consumers in this category are those organisms that depend upon plants for their food. So primary consumers are the one which mainly depend upon green plants or their parts for their food. So they depend upon the green plants. Secondary consumer are the one which depends upon primary consumers for their food. For example, fox, wolf, peacock, all the animals which eat other primary consumers would be known as secondary consumers. Okay. Tertiary consumers. In this category, those organisms are included that depend upon secondary consumers. So, secondary depend upon, just let me change the color of the pen. So, if I talk about primary consumer depend upon plants. Secondary consumer depend upon primary consumer. Tertiary consumer depends upon secondary consumers. So if I just take the example, the organism that live on green plants are primary. The organisms that depend upon green primary animals are secondary. So example is like I have fox, wolf, peacock, and the cat in this category I will have those consumers which depend upon secondary consumers for their food. The example would be I have tiger, lion, cheetah, which eats wolf, fox, peacock. So tertiary eat primary. Sorry, I'm so sorry. Tertiary eat secondary. Secondary eat primary. Primary eat the plants. Okay, and decomposers feed on dead consumers. It can be any type of dead consumer or dead producers to convert them back into physical elements. I hope there's no confusion in this and we are clear about what is ecology. It's the study of interrelationship between the living organisms and the environment, and the entire thing ecosystem can be divided into two, which is the living and the non-living. The living is the animals and the plants, and the non-living is the environment that we have. Okay, now if I talk about the abiotic component, which is the non-living part, now the examples would be water, light, temperature, air, humidity, minerals, etc. They all form a part of abiotic components. And then we have food chain. Now food chain is basically the transfer of energy from the producer to primary to secondary to tertiary. So food chain is the transfer of energy from a producer through a series of organisms. Okay. Let's talk about human blood now. Human blood. Blood is a fluid connective tissue. The quantity of blood in the human body is 7 to 8% of the total body weight. So out of the total body weight, around 7 to 8% of the weight is by the blood. The pH value of blood is 7.4. So it's a dissolution of a base whose pH value is 7.4. There's an average of 5 to 6 liters of blood in the human body. So 5 to 6 liters of blood is there in a human body, which forms 7 to 8% of the total weight. Females contain half liter blood less as compared to males. Okay. Now, blood, if I talk about, it carries the critical function of transporting oxygen and nutrients to all the other body cells and getting rid of the unwanted material such as carbon dioxide, ammonia, and the other waste products. So its purpose is to transport oxygen and nutrients and to get rid of the unwanted part that is there. Okay, blood consists of two parts, plasma and blood corpuscles. So if I talk about the plasma first, the liquid part of the blood, which forms 60% of the blood, is known as blood plasma. So 60% of the blood is formed out of the liquid part, which is known as plasma. Out of the 60%, 90% is water. So plasma is 90% water, 7% protein, 0.9% salt, and 0.1% glucose. The remaining substances are in the very low quantity. The function of plasma is the transportation of digested food, hormones, excretory products from the body. So whatever is the digested food, hormones, excretory products are transported by plasma. Okay. The second part is the blood corpuscles. Now, if I talk about the blood corpuscle, it forms the remaining 40% of the blood. So, 60% is plasma and 40% is corpuscles. Now, this is divided into three parts: RBCs, WBCs, and platelets. RBCs are red blood cells, WCs are white blood cells, and blood platelets. We'll talk about all the three parts one by one in the next slide. So let's look at RBCs first. RBCs, also known as red blood cells, carry oxygen throughout the body. So the main purpose is to carry the oxygen throughout the body. They are created in the bone marrow. So they are created in the bone marrow, and their lifespan is from 20 days to 120 days. The destruction of red blood cell take place in the liver and spleen. Therefore, liver is known as the grave of RBCs. RBCs, they die, their destruction takes place in the liver. So, liver is also known as the grave of RBCs. Okay. Now, RBCs contains hemoglobin. In which there is the word hemo, which means iron, and therefore their color is red. So it's a hemo, iron containing compound, and due to which its color is red in color, and therefore it is named as what? Hemoglobin. Okay. And their main purpose, I've already told you, is to carry oxygen throughout the body and bring back carbon dioxide. Anemia is caused by the deficiency of hemoglobin. So if hemoglobin is less, people suffer from a disease which is known as anemia. Okay, let's move forward and let's talk about WBCs, also known as white blood cells. Now, white blood cells are also known as leukocytes. They are involved in maintaining the immune system of the body. So their formation takes place in the bone marrow, basically in the lymph node, and sometimes in the liver and in the spleen. So the formation happens in the bone marrow, lymph node, spleen, and liver, and they are very, very important for maintaining the immune system of the body. Their lifespan is very low as compared to RBCs. They only live for 1 to 2 days. Nucleus is present in WBCs, and I forgot to tell you, there's no nucleus which is there in RBCs. So there's no nucleus in RBCs. there is nucleus which is present in WBCs. Okay. The ratio of RBC to WBC is 600 is to 1. So WBC and RBC, their ratio is 600 to 1. The main function of WBC is to protect the body from any disease, basically to maintain the immune system, and their main function is to carry oxygen and bring back carbon dioxide. Okay. Moving forward, platelets. They are only found in the blood of humans and other mammals. Yeah. So platelets are only found in the blood of the humans and the other mammals that we have. There is again no nucleus that you have in case of platelets. The formation takes place again in the bone marrow. The lifespan is 3 to 5 days and it dies in spleen. The blood platelets dies in spleen. The main function of platelets is to help in the clotting of blood. These blood platelets help in the clotting of the blood. Okay. Now, if I just combine all three of them, the main function that the blood carries out is to protect the body from any kind of diseases, right? To carry oxygen throughout the body and to bring back carbon dioxide, and clotting blood platelets help in the clotting of blood, and it also maintains the temperature of the body. So blood also helps in maintaining the temperature of the body. Okay. Now if I talk about the blood groups, the various blood groups that are there are A, B, AB and O. So the red blood cell type, if I talk about uh, A, B, AB and O. Now if I talk about the different blood group type, they are categorized on the basis of antigen which is present in the red blood cell. Let me just write it down. Antigen is present in red blood cell and red blood corpuscles, and on the basis of this antigen, the blood group of the human is decided. Okay. uh, if the blood contains antigen A, it will be termed as group A. If the blood contains antigen B, it will be termed as group B. If the blood contains both antigens A as well as antigen B, it will be termed as group AB. If the blood contains neither of the antigen, there is no antigen which is present, it will be termed as blood group O. Okay. So there are basically two type of antigen that we have: antigen A and antigen B. Antigen A present, group A. Antigen B present, group B. Both present, AB. None present, it will be termed as blood group O. Okay. Uh, Opposite to the antigen, there is a protein which is present in the blood, and that is known as antibody. So antigen is present in the blood, and opposite to that there's a protein which is found in the plasma, and this is known as antibody. Again, antibody are of two types: antibody A and antibody B. So now if I just look at it, group A, the red blood cell has antigen A and the plasma has an antibody which is B. It cannot be anti-A. Anti-A will destroy its own antigen A. So the antibody which is present in plasma is anti-B and antigen is A. If I talk about blood group B, the antigen which is present is antigen B. Therefore, the antibody which will be there will be A. Antibody is A. It cannot be antibody B, because then antibody will destroy its own antigen. Now, if I talk about the group AB, there are two type of antigens, A and B, which are present. So there is no antibody, because if there is an antibody, it will destroy the antigen. So there is no antibody which is present in the plasma. And if it is group O, so that means there is no antigen which is present. So there will be both the antibodies A and B would be present in the plasma of that person. You can just pause this video, look at this chart once again, so that there's no confusion about the different blood groups that we have. Okay. Now antigen A and antibody A cannot live together. Similarly, if you have antigen B and antibody B, they cannot live together. Okay, so if there's a transfusion of the blood which is done wrongly, then the blood gets spoiled, that the blood gets sticky. This is known as agglutination of blood. Therefore, whenever blood transfusion is done, it has to be very carefully done, so that there is no antigen antibody agglutination which happens. So there's no if there is antigen A, there cannot be antibody A in the blood. If there is antigen B, there cannot be antibody B in the blood that you are transfusing. Okay, so if you just look at this, blood group O is known as universal donor. It's a universal donor because it does not contain any antigen. So this is a universal donor, and because it does not contain any antigen, and AB is known as universal receptor. That means it can accept any other blood type. It is universal receptor because it does not contain any antibody. So it is universal receptor, whereas this is a universal donor. It has both the antibody, so it cannot accept any other kind of blood. If I transfuse blood group type A, anti A will destroy that. If I transfuse the blood group type B, anti B will destroy that. So O can take only O, but O can donate to everyone. So O is a universal donor, whereas AB is a universal receptor that we have. So this is about blood groups of human. The other thing that we have, so just you can look at this chart and this thing. So O is a universal donor. O can donate to O can donate to A, B, and to AB. Whereas AB is a universal recipient. It can take the blood from AB, it can take the blood from B, it can take the blood from A, it can take the blood from O. O can donate to everyone. AB can take the blood from everyone. So if I talk about A only, A can give the blood to A or AB. B can give the blood to B or AB. If I talk about AB, AB can give blood only to AB. Okay, it will not be able to give blood to any other type. Yeah. So I hope there's no confusion now that you have in this. So O is the universal donor, coming back again, and AB is the universal receptor that we have. Okay, coming to the RH factor. Now, what is this RH factor? RH factor can either be positive in the person or negative. So, in case there is RH factor which is present in the blood, it is known as RH positive. In case that factor is absent in the blood of the person, or that person is known as RH negative. Okay, so at the time of blood transfusion, RH factor is also tested. If it is RH positive is given to RH positive only. RH negative give bloods to RH negative people only. If the blood of RH positive is transmitted to RH negative, then again agglutination will happen and the person with RH negative blood group dies.